Hello, welcome to the Reflections on the History of Modeling and Theory, one of the podcast series from the new Society for Modeling and Theory in Population Biology, SMTPB. That's at smtpb.org. So the goals of the society are to provide a forum for discussing theoretical issues, tricks of the trade, and new approaches to strengthen the connection between practitioners across our field and across the world, to build a community through meetings, workshops, and other events, and also to foster the career of modelers and people who want to learn more about theory in population biology. So this particular podcast is a series in informal chats with leading figures who have really shaped our field and who can reflect and give us their insights about how modeling and theory has changed in population biology over their careers. So we are joined today by Kaylee Charlesworth. She's our technical wizard and one of the graduate students from the University of British Columbia, by John Wakely from Harvard, and we are all delighted to welcome today Brian Charlesworth from the University of Edinburgh. Brian is a professor emeritus at the University of Edinburgh. He did his undergraduate at Cambridge in natural sciences in 1966, followed by a PhD in genetics in 1969. He was a postdoctoral fellow at Chicago, followed by positions at Liverpool, then Sussex, then Chicago, before eventually moving to the University of Edinburgh as the Royal Society research professor there and the later head of the Institute of Evolutionary Biology. He's received many awards, including the Fellow of the Royal Society, Fellow of the National Academy of Sciences, and really all of the medals of high distinction from the various societies in our field, including the Darwin Medal from the Royal Society, the Frink Medal from the Zoological Society of London, the Darwin Wallace Medal of the Linnean Society, and the Thomas Hunt Morgan Medal from the Genetic Society of America. He's, I think by the Scopus count, it's always hard to know, has over 370 publications that have earned him an H index of 90, and has really shaped forget the numbers, he's really shaped the understanding that I and many people have in the field of so many subjects in population and evolutionary genetics. And that goes from understanding age-structured populations and how they are subject to selection, to understanding mating system transitions between dioici and hermaphroditism, understanding sex chromosome evolution, understanding shifts from isogamy to anisogamy, evolution with recombination, multi-locus dynamics, and really kind of discovering a new field of how molecular signatures are influenced by hitchhiking at surrounding sites as they purge deleterious mutations, a phenomenon now known as background selection. So through these, and this is just the tip of the iceberg of the many areas that Brian has contributed to our theoretical understanding of evolution. Thank you, Brian, for being with us here today.
Thank you for this flattering invitation. You seem to know more about my work than I do. But
Yes, thanks, Brian. So our first question we wanted to ask you is, when did you first realize that mathematics was such a great tool, for providing insight into biological questions?
Probably when I was in high school, embarrassingly enough. We had a genetics course, and of course genetics is inherently, as Sturtevant and Beadle said in their textbook, a mathematical science. You have a mathematical prediction about what should happen in a crossing experiment, and you go out and look and see whether that's verified. Then I got interested in evolution, and I read a book by a guy I think is totally forgotten, somebody called Carter, G.S. Carter, at Cambridge, lecturer on evolution, and he had some very elementary population genetics in it, and that kind of tweaked my interest. You know, pictures of Saurites, just stationary distribution curves, and so on. And that's so much less boring than the sort of names of the bones of skulls and that kind of stuff, which you learn in undergraduate biology, high school biology classes, and a lot of undergraduate ones as well. So that's kind of got me started.
Had you had training in mathematics?
Very little, actually. Last formal course I took when I was 16. So that was at high school. So I didn't do any maths at university at all. So I'm largely ill-educated in mathematics. I would not describe myself really as a true modeler, in the sense that that's what I started out wanting to do. I always started off thinking of myself as an experimentalist, and sort of gradually drifted into modeling in order to understand things better.
I think that's very inspiring, especially for the many students who feel like they haven't had perfect mathematical training in order to enter the field. So, I think that really opens up possibilities for many people, and you clearly have become one of the strongest theoreticians in the field.
Yeah, well, if you look at the kind of maths I do, it's pretty much high school maths, really. I mean, it's a bit of matrix algebra here and there, a bit of calculus here and there, but nothing very posh.
But isn't it part of the issue that we all learn a lot of math in our schooling, and then it only takes this much more to apply that math to something very interesting?
Yeah, I mean, I saw Wright didn't really have any strong mathematical training, to be honest, or Jim Crow, for example. So I think, or Hawley and Fisher had fairly tough mathematics courses, but I'm not trying to compare myself with them anyway. So I think if you want to figure out how to solve a problem, there are places these days it's easy to look stuff up on Wikipedia, for example. You don't even have to go to a library.
Yeah, it's those questions that really drive. Once you have a good question, then it's just a matter of looking around to find what you're approaching it.
I can't conceive of ever having been able to do anything like the work, say, Kimura did in deriving all that diffusion equation stuff back in the 50s and 60s and 70s. I mean, that's pretty hairy stuff. His solution to the general solution to the drift equation with these Gegenbaur polynomials and all that kind of fancy stuff. That's totally beyond me.
So you mentioned Carter. Were there other people who really inspired your early career research?
Yeah, well, I got a lot out of reading Douglas Faulkner's very good textbook of quantitative genetics, and he thought of quantitative genetics as population genetics and what we now think of as quantitative genetics. So it's very much as an introduction to elementary population genetics, and then the sort of quantitative trait inheritance tacked onto it. And it's a very readable book even now. I came across it in my college library when I was an undergraduate, and then we did a certain amount of quantitative genetics in the final year genetics course, which I took at Cambridge. But it wasn't very well taught, the population genetics there. It was taught by a man called George Owen, who was a PhD student of Fisher, and he'd kind of lost interest in research by the time we were there, and I'm pretty sure he gave his lectures by having a pile of papers, and he would start one year the lecture course with where he got up to last year.
Oh no!
And he spent most of his time chasing poltergeists, and actually ended up in Toronto running an institute for poltergeist research, believe it or not.
You're not joking. Real poltergeist.
Well, I don't know if there is such a thing as a real poltergeist.
No, he thought they were real. So who did you train with in your PhD?
Well, my supervisor was John Thoday, who was largely famous for being one of the first people to try and map quantitative trait loci just using conventional markers and working on analysis of disruptive selection experiments. But he was a totally hands-off supervisor. So I got much more out of interacting with the other graduate students and reading in the library. The genetics library was very good because they had reprints going all the way back to the very beginnings of genetics. Some of them signed by, the great names in genetics. There was a slip of paper in one of them which I'm very sorry not to have stolen because it was signed by Bridges Muller and Sturtevant and Morgan. I wish I'd nicked it, but I didn't.
I suspect it's not still there. We'll have to take a look.
Probably
Somebody will
not.
have to take a look.
And there was, very good PhD students there, so we talked a lot. And badmouthed our supervisors and that kind of stuff.
And I think a lot of students learn a lot more from each other than from the professors.
Yeah. Well, the tradition in Britain in those days was sort of, John Maynard's put it, you could be around, There's no real active sort of suggestion of what you should do, or, you just got on with it in some way. My PhD was pretty boring, so let's not talk about it.
Well, but to follow up on that, could you describe, or is there a particular project you were working on or moment when you had a kind of an aha for yourself that you really could solve something?
Well, that really came later when I was started as a postdoc. When I started with Dick Lewontin, I had a clue what I was going to do. And again, his philosophy was to let people be around. He would rarely tell people what to do. And he was very scrupulous, in fact, about not taking any credit for anything which his postdocs or PhD students had done independently. So I was sort of scratching my head, and I was a project, and in fact, what I did start doing was measuring linkage disequilibrium in Drosophila, because Lewontin and Franklin had come up with this kind of crazy idea that the genome was, highly in linkage disequilibrium because of strong balancing selection. And he thought this was the solution to the genetic load problem. So I thought, well, you could try and test this by measuring linkage disequilibrium between allozyme markers. It was probably somewhat optimistic, but so that was what I did experimentally for a couple of years. And then I was talking to Tim Prout, actually. He was visiting the lab, and he said something about, I don't believe in Malthusian parameters. So I said, well, what do you mean? He says, well, they're just sort of hand-waving. So I there was another person, Ted Giesel, who was working on trying to model effective size in age-structured populations. So there was I thought, well, maybe we could try and sit down and make a proper model of an age-structured population and see what comes out. And that got me into that. And I actually pretty easy. You sit down and write out the equations to show, under what conditions there is such a thing as a Malthusian parameter, and how it, how it relates to what actually happens in a population of age structure. So,
So you went just then from linkage disequilibrium to a conversation with Tim Prout to age structure. Is there a thread running through that?
Not really. I mean, the idea was to test Lewontin's theory. Tim was, as you probably know, Tim, a great character, and he was visiting, and Lewontin gave him the manuscript of this paper with Ian Franklin about the congening of the genome, and Tim comes into the lab one morning waving this manuscript saying, "Balderdash!" And so that sort of, again, can we test this idea? And in fact, when you do look for linkage disequilibrium, it's very difficult to find it between allozyme loci because they're not very closely linked. But actually, the Lewontin's theory would have predicted strong linkage disequilibrium even between loosely linked markers. So essentially, it destroyed Lewontin's theory. I wasn't the only person to do this. Mukai and Chuck Langley had also independently been estimating LD. And Deborah actually was involved in the project as well. as a sort of sideline on her postdoctoral work. So, but there was really little connection between the two, except one was purely experimental and the other was purely theoretical.
Do you, Brian, I want to know what my reading list has on it tomorrow. Do you have a favorite paper of yours that you think people don't know about, but they should?
Well, probably some I don't know about myself. The one I gave that some thought. There's a paper which I did with Chuck Langley and Wolfgang Staffen in 1986, and that was in genetics, and it said basically two things. One is a selective reason why you might have suppressed crossing over among functional highly repetitive sequences like telomeric and centromeric sequences. And basically the idea was you want these things, you need the right copy number. And so if you have too much crossing over and unequal exchange, you get the wrong copy number. So there's a selective advantage to suppressing crossing over around centromeres and telomeres. The second was, given that you've suppressed crossing over, there could be a mechanism for accumulation of more repeats in the regions of suppressed recombination, basically because if you undergo unequal exchange, you end up with an absorbing boundary where you end up with just one copy. But if you suppress recombination, you're not vulnerable to that loss of the repeats. So we thought that was kind of a neat idea, but it sort of fell flat, I would say, partly, I think, because it's beyond the comprehension of molecular biologists who can't think in population terms, as you probably have encountered this problem. And I don't think anybody in the population genetics community was interested in the problem. So it fell sharply between two stools. I thought it was a nice piece of work, and Wolfgang actually went on and did some deeper theoretical modeling of it and showed it really does work as far as the repetitive sequences accumulation in low recombination regions are concerned. And of course, the empirical evidence for that is overwhelmingly strong that this is actually what happens. But it seems to pass. Yeah. World passed it by, but you're welcome to go out and advertise it to all your friends.
Well, and you know, this is only one of many of your papers where it really preceded the major empirical effort or an expansion of our understanding. And so centromeres have been studied substantially in the last 10 years in our understanding of repeat evolution in centromeres. So I like this idea that the reduced recombination is really needed in the centromeric regions to avoid collapse of the number of repeats.
We also pointed out that there was evidence that if you have crossovers near centromeres, this causes non-disjunction. This was actually work done by a chap called Ed Garber at Chicago. He was another interesting character, actually. And in Astelago, which is now called microbotrium, he pioneered the genetics of that system, in fact. And he had a nice paper on that. And this is now widely accepted in human genetics and fly genetics that crossovers near centromeres can cause non-disjunction, and the mechanism is quite well understood. He didn't know what the mechanism was, obviously.
At the time, right. Thank you.
So it's another reason for suppressing crossing over near centromeres. You end up with the wrong number of chromosomes at some rather small but measurable frequency.
Right.
So, Brian, today it seems, as much as ever, there's a role for theory in biology and genetics and evolutionary biology. Do you have any advice for people who are just entering the field now, just getting started?
Well, yeah, I mean, you might go and work for a hedge fund. No, well, if you want to, I mean, you just have to have I think there are lots and lots of problems. I don't think there's a problem in finding a problem. It's just I think the main thing is to try and find something interesting, which hasn't been done before and which you can actually do. I mean, that's one of the big problems in science, is finding something that's both interesting and doable. We had a faculty member in the genetics department, John Pateman, his view of science was, well, everything that's worth doing has been done, and everything everything that can be done has been done, and everything that's worth doing can't be done. That's really not good advice to give to beginning science. It isn't even true, but it's kind of a it's a lucky dip in some sense, finding stuff which is both interesting and actually doable, especially within the course of a PhD. I mean, I think a lot of PhD students come to grief because they just end up with something which looks very interesting, but they can't really do anything much with it, or else they can do something with it. It's really boring, and they get browned off by the problem. So, but I don't have any real advice on how to find something which is worth doing and doable. It's kind of a bit of a lucky dip, I think.
Yeah, that seems true any time. I'm wondering if there are ways you think things have changed, nowadays versus previously.
Oh, I think it's a lot more difficult now, certainly in the sort of population and evolutionary genetics era, because with all the expansion of sequencing and stuff, it's not good enough for somebody to sit down and be able to scribble some algebra. They also have to know all this stuff about bioinformatics and sequencing technology, etc. I mean, it's totally beyond me, and I am always amazed at what my colleagues can do in that respect. And there's a huge range of different skills I think you need now. I mean, in my day, genetics was largely a matter of looking down a microscope and pushing some flies, or looking at salivary chromosome bands, or worst case scenarios, running electrophoresis gels in the lab, which even I could learn how to run a gel with a few occasional lab mishaps due to absent-mindedness. But now, I mean, it's very demanding, I think. You have to have a broad range of skills, and it takes a lot of energy and time to acquire those. So yeah, I think it's a lot more difficult. Yeah.
I agree. I want to go back and to the point about how do you identify those good questions. But one of the things that really stood out, I think, in your career is just how closely empirical work and theory were.
Yes, well, I spent a fair amount of my time doing work in the lab, actually. I mean, probably more time than doing the theory. I used to do a lot of work with flies, breeding experiments and counting flies and looking at salivary gland chromosomes. I haven't done any of that for years now. My back is kind of giving out, so I really lost the appetite for sitting on a bench for hours on end. But I think it's good to do that in some ways, because one of the problems of being a real theoretician is you've got to have a exaggerated respect for the quality of the data. When you actually collect data yourself, you know what all the kinds of problems that are lurking there, and when you do an experiment, you're never quite sure. Well, is this result really true, or is it just some kind of screw up somewhere which I haven't thought about? At least that's always been my worry. I mean.
Yeah, that makes sense. And to some extent, the questions that you then are like, yeah, could this be something else? You can address sometimes more easily through theory by changing all of the possibilities and seeing if it still will work.
You mentioned collaboration, I think, as something that it may be incumbent upon new theoreticians to do. Are there other
Yeah.
ways you think that things have changed?
Well, computation, of course, computational power is ginormous compared with what was available when I was starting. I mean, Deborah and I were among the first graduate students at Cambridge to use a computer to analyze our data. and mostly analyses of variance programs. And by God, was that tough. The computer was located in the University Mathematical Laboratory, which was about a half-an-hour bicycle ride from the genetics department. And the way it worked is you would type in your code on punched tape, and you'd in the what they call flexi writers in the mathematical lab, and then you feed these things in, and the machine would process it. It probably had the about 1/20th the power of a mobile phone, or 1/200th, I don't know. And then you'd cycle back, and then you come back the next day and look for your output. And of course, usually when you write a program, it's all full of errors, so it would take days to actually get something to work.
I'm surprised you keep at it after punching all
I
those
mean, it was a
cards.
nightmare. I mean, it got better with punched cards. That was quite a pleasant advance, but terminals didn't come in till mid-80s. I mean, we didn't have terminals at the University of Sussex, remote terminals, till about 1984, and that was kind of bliss in a way, because it's so much easier to write and debug a program than when you had to fool around with punch cards. And the worst with punch cards, you go down the stairs, you trip and drop the whole bloody pile, and then you had to spend hours sorting them into the right order.
I think that's an ongoing issue, isn't it? I was just in preparation for this morning reading your paper on background selection, 1993, and you have a line in it. It says right here, the program was written in the C language for a Macintosh 2 computer with a Tektronix RP88 coprocessor, which probably was like
Yeah,
a very
right.
fancy thing at that moment.
It was quite good, actually. Yeah. Yeah, that was Martin Morgan actually did the programming. It was nothing to do with me. Yeah, no, the desktop computers were just kind of bliss compared to that crappy old technology. And they were just as powerful as a mainframe by then. I mean, an old-style mainframe, obviously not a supercomputer or anything. So that's been a huge change. It's a double-edged sword in some ways, I think, because now, there are all these sort of off-the-shelf simulation packages like Slim. So now, enthusiastic amateurs, as you might say, can get into doing simulations, and without actually a guiding framework, I think simulation can be a really dangerous thing to do, because you don't know what to expect, so you take results at face value. You don't really know how to interpret them, etc. So I firmly believe if you're going to do simulations, you should have some idea from some rough theory that what's going to come out of it at the other end. Otherwise, you can run into trouble. I won't name any names.
This is
Thank
being
you.
recorded.
Yeah, right.
One of the things that I feel has changed over the last century is just how subdivided biology is, where you're getting more and more trained in molecular genetics or biochemistry or population evolutionary population biology or community ecology, population ecology. Anyway, how does the splintering of fields, how do you think that has influenced theory for the good or the bad?
Well, the good stuff is, of course, with the molecular revolution, you can actually apply classical population genetics theory to the real world. Before you could look at DNA sequence variants, there was limited areas in which you could analyze selection acting on inversions, or study sickle cell hemoglobin and that kind of stuff, based on Bachelararia, the usual suspects. But to apply, for example, the infinite sites model of molecular variation, until we had reasonably good base of DNA sequence polymorphism data, there really wasn't much you could do with that theory. There's that famous passage in Dick Lewontin's 1974 book about the elaborate machinery which has been constructed by the theorists, and you fed in the electrophoretic data, and there's a sort of clashing of gears, and nothing much came out the other end. And so now the field of population genetics, at least, has got the ultimate data. I mean, there's nowhere else to go except DNA sequencer data. And if we can't do a good fist of analyzing that, well, we might as well pack up our bags and retire to Florida or somewhere. So that's I think a huge benefit. The downside, maybe, is there's just so much data we don't know what to do with it. And as you say, it requires so many different skills, computational, technological, etc. It's getting into a situation like physics, really, where it requires teams of people to actually produce a single paper, and that's fine. Except I think it can be a bit demoralizing for a lot of people involved. You see all these genome sequence papers with God knows how many people on it, and one of them is like some guy at the Large Hadron Collider who's responsible for tightening the bolts on the magnets or something. It's you're not feeling you're seeing a broad picture. I think it must be. I don't know what the people involved in these big teams actually feel about this kind of work, but it's.
Any advice that you could give people starting out about how to keep that broad picture in mind, or just to know enough about different areas to synthesize?
Well, I think something which strikes me is that a lot of people don't actually try and read the classic literature these days. They sort of assume that something which was published more than, say, 5 years ago is kind of irrelevant. And I think one thing is a good thing for PhD programs, and some of them do. I know we did it at Chicago, and I know other people do it. Is to encourage students to read classic papers and get a broader picture of where the field has come from, and realize that not everything was discovered in the last 5 weeks or whatever. That, some very important things which you should know, which were discovered a long time ago. One of the nice things about Drosophila genetics, as opposed to population genetics, is that the people, even at the absolute cutting edge of the field, people like Scott Hawley, have enormous respect for the old masters, Bridges, Marlowe, Sturtevant, and those guys. Whereas I think people in other branches tend to forget there's a history of the field, and they think that's the latest thing is the only thing which matters. And I think in PhD programs it's important for students to get a good perspective on where the field has come from, as well as of course knowing what's going on at the present. It's a hard act because there's so much now. I can understand where the problem arises, but
well, that leads very well, I think, into what will be the final question of this interview. It really comes through in your work, the constant reference back to classical population genetic results and work. And you have initiated a number of what turned out to be very fruitful lines of research with lasting importance. Many of the theoretical things you've done underpin analyses of data today, from molecular data, signals of selection, and so on, to properties of age-structured populations. And I'm just wondering if you could talk a little bit about how and whether you saw these things at the time as, new areas for research, and how it allowed you to see these as good problems to work on.
I'm not one of these people who has a sort of organized plan of what I'm going to do next, really. I sort of kind of wander into things. And I mean, we discovered background selection by the back door, so to speak, which was Martin Morgan was running simulations of a modifier of the rate of selfing introduced onto a background of deleterious mutation. And we thought we'd just check that everything was working properly. But instead of putting a modifier with an effect, it just put a neutral mutation in to see whether it followed, the rules which neutral mutations were supposed to follow. And we discovered they didn't, that the Sojourn time in particular was considerably less than what the, diffusion equation result predicted. So I thought, oh, well, we screwed it up somewhere, so we checked it all through and it looked fine. And suddenly a sort of light bulb went on. Well, actually, yeah, maybe the deleterious mutations are doing something which pulls down the effective population size. And that's why it's not obeying the rules, which of course started thinking about that. So, it was entirely serendipitous, I think, to be honest. It wasn't something we set out to discover, but we had an observation. And I think this is actually quite common in science. You find something which doesn't quite fit. So you think, well, something going on there. I need to try and understand what it is. So, it's kind of problem solving, really. Something gets your attention. And I think I think somebody said, tragedy or exceptions. Maybe it was Sturtevant. And, something which looks funny. Let's try and think it through. Or the other thing which also happens is the people accept things. When we started out, breeding system evolution was all for the good of the species. It's absolute dogma. And, just to think about it for a few minutes, even when I was an undergraduate, I thought this doesn't really make a lot of sense. But I didn't, Deborah and I didn't do anything much about it. But later on, we started thinking, well, you can actually do things which don't require the good of the species. And I think it wasn't just us, there were quite a lot of other people involved. The whole tenor of work in mating system evolution has changed completely since the 70s, when the good of the species, which is still, I think, quite a concept dear to a lot of molecular biologists, but it's vanished almost entirely from evolutionary biology in the naive sort of way. I mean, there's still arguments about how important group versus kin selection is, but it's conducted at a totally different level. So, I mean, looking for anomalies and challenging accepted thinking, I think, are good ways to find things which are worth doing. Obviously, you can go too far. There are irritating people who are constantly trying to create a new paradigm, but there's always scope for cracks to be filled in the edifice of science, and there are things which people have overlooked. Everybody makes mistakes, and so I think that's a way forward, is to look for those cracks and anomalies and try and deal with them.
Thank you so much, Brian. Is there anything else that you would like, any parting words of wisdom or anything else we haven't asked yet?
Honestly, we seem to have covered a fair bit of ground. I'm not sure
We have.
Got words of wisdom. I mean, I would certainly like to think that we've got lots of enthusiastic young people coming into the field, but I realize that it's got ever increasingly difficult for people to get proper jobs in science and that kind of stuff. But I hope they'll have the courage to face up to the difficulties.
Well, I think it's inspiring to learn about careers like yours, where, without a strong math background, that didn't keep you from becoming one of the most influential theoreticians of our age. So thank you for this conversation today.
Well, thank you a lot. It's been a lot of fun.